This is the polymerase chain reaction, which is an immunological test, and we're just going to go over some of the facts about it. All right, so the history
of the test.
All right, so in 1985, a scientist named Carey Mullis was working for the CETIS Corporation, which is a company based in California. The company no longer exists now, but at the time he was a chemist working there, and he needed a way to replicate a very small amount of DNA and create a lot of it. So he wanted to have millions of copies of just a single strand of DNA. So he developed this technique that involved repeatedly heating and cooling a solution of the DNA along with some nucleotides. And basically they would adapt to the shape of the DNA, and when you cool it, they'd come out, and you'd have a bunch of copies of the DNA that you could repeat over and over again. And this was a really groundbreaking solution that Mullis later went on to receive a Nobel Prize for his work.
Next, the key people involved.
Yeah, so for the key people involved, again, it's just Mullis with his PCR technique, and he just wanted to create large batches of the DNA. And the work had many applications in immunology and AIDS. For diagnosis, it was a replacement for some of the old tests that they did, and PCR was much better. And Carey Mullis actually recently died on August 7th in 2019 in California.
And now the key principles involved in PCR. So there are 3 steps in PCR, but the ingredients needed for this are Taq polymerase, primers, template DNA, and nucleotides. So the first step is denaturing, and that's when you heat the DNA strands so that they will result in single strands. And then the next is annealing. So the strands will be cooled so the primers could bind to the complementary sequence on the single-stranded template DNA. And the third step was extension. So the temperature needed to be raised so the Taq polymerase could extend the primers and synthesize new strands of DNA. So basically it's just copying the DNA template a bunch of times. And you can see the result of this by a technique in which fragments of DNA are pulled through a gel matrix by an electric current, and it separates DNA fragments according to size. Next.
Oh, used for the first time. So the history of the PCR, it was used in 1983 by scientists at the CETIS Corporation, like Raj mentioned, came up with the PCR as a method to copy DNA and synthesize large amounts of specific target DNA. Okay, cool. For the next 2 years, a team of scientists recognized the future impact of the PCR on molecular biology. They researched, refined, and made the theoretical process a reality. The team presented the PCR publicly for the first time in 1985 at the American Society for Human Genetics annual meeting.
And present-day uses of PCR. The first one is genotyping. qPCR is used to identify a single nucleotide and involves designing primers specific to the mutation or allele being studied. In cloning, PCR is used to clone DNA fragments in large amounts. In mutation detection, PCR makes hundreds of different copies which can be analyzed for mutations. In sequencing, DNA segments can be amplified to 1 million-fold to iterate through them. And it is also used in microarrays, forensics, and paternity testing. And companies that use PCR.
Yeah, these are just some of the companies that are involved in the location of the actual plant that does the PCR, managing the PCR solutions that they receive, and then evaluating or evaluations of the PCR solutions that they have. So a couple of them are GNA Biosolutions, IQBay, and QuantaBio. And a lot of these companies are primarily based in California, the northeastern United States, and the New York metro area, or in Boston, or in markets in Europe and China. And there are a couple of organizations that aren't companies involved with PCR, like the CDC and the European Institute for Genetics. And yeah, these are just some of the organizations involved.
COVID-19, can it be used for COVID-19 testing? Yes, it can, because in conjunction with antibody testing, there are two most common ways for detecting coronavirus. A person's DNA is amplified using PCR to detect if the virus is in the DNA of their cells. RT-PCR is used to turn the RNA of COVID-19 into DNA, then replicate it to create the whole genome from 100 nucleotide sequences. If both genes detected positive, if one inconclusive, and if neither test, it's negative.